The overall goal of this procedure is to isolate subpopulations of human adipose-derived stem cells with enhanced osteogenic potential using flow-assisted cytometry. To accomplish this, human adipose-derived stem cells, or ASCs, are harvested from human lipoaspirate and cultured for 36 hours. The cells are then stained with antibodies selected to label pro-osteogenic populations, and the desired populations are isolated using flow-assisted cytometry. Ultimately, alkaline phosphatase and alizarin staining reveal enhanced osteogenic potential of the isolated cells.
The implications of this technique extend towards regenerative medicine and the development of new translational therapies because it describes how to isolate a subpopulation of ASCs with enhanced osteogenesis.
The following procedure is performed using human subcutaneous adipose tissue from healthy female patients who have undergone elective lipoaspiration of the abdomen, flank, and/or thigh region under local or general anesthesia. Begin with 500 milliliters to 1 liter of tissue in a suction canister taken directly from the operating room. Immediately upon collection, begin the harvesting procedure. This will maximize the cell viability. To obtain the stromal vascular fraction, or SVF, from the lipoaspirated adipose tissue, begin by adding sterile PBS at a ratio of 1:1. Then, gently shake the canister to mix. After mixing, allow the solution to stand until it separates into 3 layers. The top oil layer